My name's Eric Fried. I'm director of the HIV Dynamics and Replication Program at the NCI in Frederick, and it's a pleasure to introduce the 2023 George Khoury Memorial Lecture. So a few words about George Khoury. George was a graduate of Princeton University and Harvard Medical School. In 1976, he became head of the virus tumor biology section in the Laboratory of Molecular Virology of the NCI. He became chief of that laboratory in 1980. The next year, in 1981, he received the Arthur S. Fleming Award for Outstanding Government Service. And in 1987, the year of his untimely death at age 43, was elected member of the National Academy of Sciences. And this is a photograph of George Khoury on the left and his mentor Malcolm Martin on the right, and a very young Peter Howley in the middle receiving the Wally Rowe Award. George published over 140 papers during his short career deciphering mechanisms of eukaryotic transcription, and this got completely garbled in the transfer from Mac to PC, but it's just a bunch of landmark papers that George published during his career. In addition to being a highly productive scientist, George had a number of highly distinguished trainees and collaborators. Many of you will recognize the individuals on this list. The George Khoury Lectures began in 1995 and featured a similarly distinguished group of speakers, several of whom received the Nobel Prize. The most recent speakers were Julie Lieberman, Julie Pfeiffer, Carla Kierkegaard, and Yuan Chang. So it's a real pleasure to introduce the 2023 Khoury Lecturer, Dr. Britt Glaunsinger. Britt is a native of Arizona. She received her bachelor's degree from the University of Arizona. She did her Ph.D. research at Baylor and then a postdoc at UCSF with Don Ganem. In 2006, she was recruited to an assistant professor position at Berkeley in the Department of Plant and Microbial Biology. She received tenure in 2012. In 2015, became an HHMI investigator, full professor in 2017, and since 2021 has been serving as associate chair of that department at Berkeley. She's received a number of honors and recognitions, too many to list here, but a few notable ones. She received the W.M. Keck Foundation Distinguished Young Scholar Award, the UC Berkeley Graduate Assembly Faculty Mentor Award for Excellence in Graduate Mentorship, and in 2019 was elected Fellow of the American Academy of Microbiology. Her research focuses on several key areas, including mechanisms and consequences of virus-induced mRNA degradation, novel modes of transcriptional regulation during late stages of infection, and viral activation and co-option of non-coding RNA. Britt's seminar today is entitled Controlling the Message, How Herpes Viruses Manipulate the Gene Expression Landscape. And before Britt gives her seminar, we discovered at dinner last night that Britt had never heard of Old Bay. So as a souvenir from Maryland and a token of our appreciation for her visit and lecture, I'd like to present her with this small tin of genuine Maryland Old Bay. Britt, it's all yours.
It's a real pleasure to be here. The flowers are in bloom. It's a beautiful sunny day compared to the rainstorm that I left in California yesterday. And as Eric alluded to, I've really spent pretty much my entire career studying virus-host interactions that relate to gene regulation. And so it is, I have to say, a particular honor to be invited to give this memorial lecture for George Khoury. In particular, I am familiar very much so with the research that he had carried out related to using DNA viruses in particular to dissect fundamental principles of gene regulation. And much of his research actually provided the foundation of knowledge that underlies many of the projects in my own lab now. So I feel like it's a real delight to be able to participate in this lecture series. When I think about viral infection, really gene regulation and the ability to manipulate gene regulation is at the crux of whether or not an infection is going to take over or peter out. And this is because from the host side, the ability to elicit a particular gene expression program in response to infection is going to determine whether or not the host cell can counteract and fight back against the viral infection, and conversely, the ability of the virus to commandeer those gene expression machinery and shut down the aspects of the gene expression that are involved in restricting viral infection really determines whether or not that viral infection is going to take off. So restructuring the host gene expression environment is of critical importance for viruses, and this is something that I've been interested in for a long time. Because this is of central importance to biology, many viruses encode proteins with the dedicated job of shutting down host gene expression. These are called host shutoff factors. They've really captivated me for years, and the idea here, some of which are shown in this diagram, is that expression of these proteins, many of which target RNA directly, leads to a large-scale downregulation of host gene expression during infection. And this does at least two different things for the virus. First, it helps the virus shunt translation resources away from the host and towards the virus, because no virus, of course, can encode the translation machinery that all viruses need. Second, by casting a wide net in targeting most of the messenger RNA in a cell, within that net, the virus is able to capture many of the immune stimulatory factors that the cell is producing in an attempt to stop the virus. And so in this regard, these viruses, by globally shutting down host gene expression, can help counteract many innate immune responses. We study several of these host shutoff factors, but really the main viruses my lab is focused on are in herpesviruses, and that's going to be the topic of the story today. Herpesviruses, I would argue, and many people in this room would probably agree with me, are among, if not the most successful pathogens on the planet, and that's in part because they're ubiquitous within the human population. Of course, once you're infected with herpes, you can not clear that infection because of their ability to establish lifelong latency. But it's not just humans. Herpesviruses are incredibly widespread on the planet. Many, perhaps most vertebrates have herpesviruses associated with them, and even some invertebrates like oysters and coral have herpesviruses. So they're widespread. They're also very ancient. It's estimated they arose somewhere between 200 and 400 million years ago, which is a really extensive coevolutionary history that they have with us and their other hosts. And this is one of the reasons why we think they make excellent models to dissect gene regulation in mammalian cells under stressful conditions like pathogen invasion. My lab is focused on a subfamily of herpesviruses, the gamma herpesviruses, that have two human pathogens, Epstein-Barr virus, or EBV, which of course causes a number of B-cell lymphomas, also recently identified as a main cause of multiple sclerosis, Kaposi sarcoma-associated herpesvirus, which is a predominant cause of cancer in AIDS patients. We also work on a mouse herpesvirus called murine gamma herpesvirus 68, which is a really commonly used model in the field, both for in vivo biology and for studying the lytic or active cycle of these viruses, which is the phase of the viral life cycle that we're really interested in. So during lytic replication, which is when these double-stranded DNA viruses are amplifying and spreading within a host, they are exhibiting this host shutoff phenotype. All of these gamma herpesviruses encode a host shutoff factor, which in KSHV is called SOX, for shutoff and exonuclease, that our lab and other labs over the years has shown the ability of this nuclease to elicit widespread RNA decay plays a variety of roles in the gamma herpesvirus life cycle, from promoting immune evasion, as I discussed earlier, to enabling the virus to traffic where it needs to go in the in vivo model in a mouse, establishing latency, being able to replicate in B cells, which are the main niche for gamma herpesviruses, and also beyond controlling the host gene expression environment, this setting the balance of the appropriate levels of host genes versus viral genes influences viral gene expression, protein production, and actually what gets packaged into nascent viral particles, which can influence new rounds of infection. So a variety of stages of the viral life cycle are influenced by this ability to control the gene expression cascade. And we've studied this process for a number of years, in part studying the RNA targeting mechanisms terms of these enzymes, which we've shown that the SOX protein can broadly target cellular messenger RNA. It targets only Pol2 transcribed messenger RNA. And although it can broadly target RNA, it does have specificity built into it. It recognizes and cleaves its RNA targets through a combination of sequence and structure, causing an internal cleavage event, which basically what that's doing is creating unprotected free ends of the RNA that can then be attacked by the resident existing nucleases that our own cells normally use to control RNA turnover. And so this two-step process of the virus inducing an internal cleavage and then relying on cellular enzymes to clear the RNA is important. And so hold that thought in your mind. I'm going to come back and touch on it in a little later in the lecture. And so beyond studying how these enzymes work in targeting RNA and what sorts of RNA features are required for targeting or escape, in the last several years, we've shifted our focus somewhat to saying, can we use these as tools to probe how cells sense and respond to large changes in messenger RNA abundance? Because these are features that are characteristic of a number of pathogenic situations. So I'm going to tell you two sort of related stories today. The first story focuses on the host side and just that question that I outlined of using these viruses as a tool to explore connections between seemingly very distal stages of the viral life cycle and how those are interconnected and the cell uses those in response to stress. And then for the second part of my talk, I'm going to flip the page and focus on the virus itself and how it benefits from this and is able to orchestrate sort of an unusual gene expression cascade or regulation for the viral genes. All right, so this first part of the story begins with the idea that cells in their uninfected state basically are trying to achieve homeostasis. They want an appropriate overall balance of messenger RNA. And this idea of cells trying to achieve homeostasis by controlling their messenger RNA levels is now well established in yeast and also in mammalian cells, where it's been shown, for example, that if you decrease the overall rate of messenger RNA synthesis, for example, using slow RNA polymerase II, the cell will respond to that and compensate for it by generally stabilizing the messages that exist. If fewer are being produced, you need those that are there to stick around longer. And conversely, if you slow down the degradation of the messages in the cell by getting rid of key decay factors, for example, the cell will compensate by slowing the rate of transcription because you've got those there sticking around longer. You don't need to produce as many to achieve your optimal balance. But we wondered, what about if you were looking in the other direction? Instead of stabilizing RNA, a much more common situation in viral infection is, as I mentioned, global destabilization of RNA. And so in this regard, would the cell also try and maintain homeostasis? We thought not, right? Cells generally, when faced with infection, are not trying to maintain the status quo. They're often trying to shut things down, as we see in translational control with PKR activation and things like that. And indeed, what we found is that in the case of cellular threat, for example, these herpesvirus infections that elicit RNA decay, the balance is tipped away from homeostasis and instead in the opposite direction, where the cell, instead of responding by amplifying transcription, responds by shutting down transcription, essentially magnifying the shutdown of gene expression in the cell. And we hypothesize that one of the reasons the cell is doing this is in part because many viruses, like DNA viruses, are reliant on the cell's machinery. And so usually cells, when they sense that they're infected, are trying to find ways to shut down core cellular processes, as I said, translation being a very well-characterized one, but we think transcription is another one, particularly for DNA viruses. All right, so let me show you what I mean by this. We first observed this connection between RNA decay and transcription using the MHV68 system, in which we had two viruses that we were using. We were using a wild-type virus, which has a functional SOX protein, or it's called musox for murine SOX and MHV68. When you infect cells, and we're using mouse fibroblast cells for this, when you infect cells, the virus induces widespread cytoplasmic messenger RNA degradation. Conversely, we can infect with a single point mutant version of this virus that we have that has a point mutation within the SOX gene that selectively inhibits the ability of that protein to effectively target mRNAs for decay. So importantly, in these cells, both viruses replicate similarly, but the difference being that the SOX mutant does not accelerate RNA decay in the cytoplasm, whereas the wild-type version does. We can then look and see how does the cell respond to either accelerated RNA decay or normal RNA decay in the face of infection at the transcriptional level. And we look at this either by looking at nascent RNA transcription using things like 4-thiouridine pulse labeling, or we look at RNA polymerase II occupancy across the genome by doing chromatin immunoprecipitation. The latter is the experiment that I'm going to show you today. And so what we observed was that, of course, in an uninfected cell, when you look at the pileup of polymerase, you see it centered across the mammalian genome near the transcription start site, as you would expect. However, during infection with wild-type virus, there's a large-scale clearance of the polymerase from the host genome, and this clearance is linked to cytoplasmic messenger RNA decay because we see that largely this does not happen if we're infecting with the mutant virus that replicates fine but cannot elicit cytoplasmic RNA decay. So what this tells us is that there is something about RNA decay in the cytoplasm that is causing a reduction in RNA synthesis in the nucleus. And so how might that happen? There are a couple of possibilities. One is which it's something about targeting transcripts individually that matter. For example, if you have factors that encode transcriptional regulators and you're getting rid of their transcripts and those proteins are labile, you might be changing the levels of proteins in the cell that are responsible for transcription and dampening transcription that way. Alternatively, or in addition, you could have a situation where the cell is actually somehow sensing the degradation of these cleaved fragments, sort of the act of RNA decay, and responding to that. And what I'm going to tell you today is that both of these things turn out to be true, and that we discovered them in sort of reverse order. So the one that we hit on first was this idea that cells could actually sense accelerated RNA decay, and the reason we knew this was the case was that if we were using a minimalist system, we could recapitulate these results in uninfected cells if we just expressed the viral nuclease, which caused RNA decay in the outside of infection. And there we knew that if we prevented degradation of these cleaved fragments, you no longer had this transcriptional repression. So it was something post-translational inactivation, but actually involving the detection somehow of the degrading RNAs. And so we set out to figure out, how does that happen? So in thinking about this, we consider what happens to an RNA as it's being degraded that the cell might be able to sense. And one of the hypotheses we ended up testing was this idea that, of course, cells the RNA in a cell is not naked. It's coated by suites of RNA binding proteins. And so as these RNAs are being cleaved and the subsequent cleavage products are being destroyed, what is happening is that these RNA binding proteins are being evicted from the RNA as it's undergoing degradation. This happens in our cells all the time at some basal rate, right, as your RNAs are undergoing turnover. But you can envision that if you are greatly ramping up the rate with which RNAs are being destroyed, you have lots more RNA binding proteins being released from these RNAs. And we know that RNA binding proteins, many of them are shuttling proteins that move in and out of the nucleus, depending as messenger RNA itself starts in one compartment and ends in the other. So we considered the possibility that perhaps release of RNA binding proteins was what was being sensed by the cell and leading to this transcriptional response, perhaps because they are moving within the cell differentially, depending on whether they are bound to RNA or released from RNA. So we tested this hypothesis in collaboration with Ileana Cristea's lab by analyzing the protein distribution and redistribution that happens in the cell in response to RNA decay. All right? The first time we did this experiment, we did it in the minimalist system where we're just trying to isolate the RNA decay phenotype. So we were taking hex cells that we had expressed just the viral nuclease out of the context of infection. And we either had a wild-type version of the nuclease, we had a catalytically dead version of the nuclease, and as controls, I told you that what we knew was that we needed this destruction of those cleaved RNAs to release the RNA binding proteins, and that was carried out by the cellular nuclease XRN1. So we engineered cells to be knocked out for XRN1 to slow that rate of decay of the already cleaved transcripts. Each of these cells we fractionated into the nuclear or cytoplasmic fraction, extracted total protein, which were then labeled with isobaric or tandem mass tags and identified by mass spectrometry. And so the idea here was we were looking for, first of all, you could look for changes in global protein abundance, or we could look for protein redistribution that would happen. And the signature for that would be the abundance of a protein would go up in one compartment and down in a compensatory way in another compartment. So for example, if we're looking for proteins that are moving from the cytoplasm to the nucleus, you'd see an increase in their nuclear signal and a decrease in their cytoplasmic signal under a given condition. And so when we did this, as it turned out, that was the dominant phenotype, that what we saw was a clear signature in cells expressing the wild-type nuclease of proteins that were shifting their distribution from the cytoplasm of the cell to the nucleus of the cell. And so what I'm showing you here are these results in a heat map in which you can see that compared to the catalytically inactive version of the nuclease, there is a set of proteins, there are about 30 or so of them, that reproducibly accumulate in the nucleus of these cells. And about 70% of those only accumulate in cells that have XRN1, but no longer in cells lacking XRN1. And we looked at all proteins, but again, when these proteins that shift, we do a go-term analysis for them. Really the only signature that pops out are things related to messenger RNA binding. So again, RNA binding protein signatures like poly-A binding, poly-U binding, messenger RNA 3' UTR binding, which are exactly the sorts of proteins that you would expect to be released from a degrading RNA that are then, once they're released, transiting to the nucleus. So what this tells us is that indeed, RNA destruction in the cytoplasm, if you ramp that up, this causes release of RNA binding proteins, which then shuttle to the nucleus. This correlates with a reduction in overall Pol-II occupancy on the host genome and transcription, but it doesn't yet tell us whether the redistribution of those RNA binding proteins is necessary to convey that signal of cytoplasmic RNA decay to transcription. And so to test that, his former student Chris Duncan Lewis used now infamous drug ivermectin, which does not target COVID effectively, but does target the nuclear import machinery, the important alpha-beta complex, which is important for relocalizing proteins to the nucleus. And so what he could see is that in cells where he treated with MOX, so like a DMSO treatment, that expressed the musox protein. Now what we're looking at is Pol-II occupancy here at a couple of representative host promoters. You can see that in cells expressing musox, you have this characteristic decrease in Pol-II occupancy because of cytoplasmic RNA decay. However, if he blocks this RNA transport, RNA binding protein transport by treating the cells with ivermectin, he'd shown that these cells are still undergoing the same level of RNA decay in the cytoplasm, but that no longer causes this transcriptional repression. So that suggests to us that indeed the movement of RNA binding proteins is the signal that is somehow read out by the cell to trigger this transcriptional repression. So one of the things that we're really interested in now is, well, this is kind of a long list of proteins here that are shuttling. And is it that individual proteins within this list are sort of key players, either contacting or signaling things in the nucleus to elicit this transcriptional repression response? Or instead, is it the mass action of generally redistributing the RNA binding proteins that is causing some sort of a stress signal in the cell that leads to transcriptional repression? We favor the key player hypothesis, and the reason for that is that we're now systematically going through and doing things like knockout and overexpression experiments for these individual RNA binding proteins. And we found a couple of examples where proteins, individual proteins, like the poly-A binding proteins, for example, seem to play important roles, whereas others, where I have arrows here, I'm not going to show you the data for because they showed no phenotype when we did these experiments. So let me just show you the example of the poly-A binding protein result. So poly-A binding proteins, cytoplasmic poly-A binding proteins, are actually proteins we've studied for a long time. These are proteins, as their name suggests, bind to the poly-A tails of messenger RNAs and play a number of important roles in RNA stability and translational competence in the cytoplasm. They're also proteins that we know from herpesvirus work and from a number of other viruses tend to get relocalized and accumulate in the nucleus in response to large RNA changes in the cytoplasm that occur during infection. So this is not a phenotype that we see only in herpesvirus infection, only in gamma herpesvirus, also seen in alpha herpesviruses and other viruses as well. So what we see here is that in the presence of normal levels of the poly-A binding protein, again, expression of the SOX protein causes this strong transcriptional repression. However, if we deplete the poly-A binding proteins from cells, now you see that this phenotype is reversed, suggesting that they are playing a role in this repression phenotype. We can also ask, well, what happens if you just drive these proteins into the nucleus in the absence of RNA decay in the cytoplasm? And we can do this through a couple of strategies, but one of which is that we, a number of years ago, had shown that the reason that poly-A binding proteins, the cytoplasmic poly-A binding proteins, are retained in the cytoplasm is that they have a nuclear localization signal that's hidden in their RNA recognition motifs. So when they're bound to poly-A tails, that nuclear localization signal is masked and they're kept in the cytoplasm. As an RNA is destroyed and they're released, that nuclear localization signal is exposed, binds important alpha, and is transported to the nucleus. So we found that if you just express, it turns out PAB its levels are pretty carefully titrated in the cell, and if you express it, overexpress it, anything that's not bound to poly-A tails just goes into the nucleus because its NLS is exposed. So when we do this, in the absence of cytoplasmic mRNA decay, if you just overexpress PAB-C on its own, increasing its concentration in the nucleus causes a decrease in Pol2 occupancy at these cellular promoters. And so for this reason that we think that these proteins, as well as we suspect a couple of others on this list, may be key players in mediating this transcriptional repression. And so now the question is, how are they doing that? And this is really an ongoing piece of work, and I'm just going to share with you my favorite hypothesis, which we're in the process of testing, that I don't know if it's going to be true or not. But this is a hypothesis generated by one of my current graduate students, Sam Ryder, who came up with the idea that perhaps what happens when you have an influx of these RNA binding proteins is that they may compete with resident nuclear RNA binding proteins, many of which play important roles in co-transcriptional RNA processing activities that happen during the process of transcription. And indeed, even the process of transcription helps compete off chromatin modifiers and things from the DNA that makes a locus transcriptionally competent. So she hypothesizes that as these RNA binding proteins go into the nucleus, that they may reoccupy sort of the space that the normal RNA binding proteins in the nucleus occupy on nascent RNA, and therefore disrupt that RNA very early on in the transcription process and alter transcription. And so what she's doing is she's in the process of purifying nascent RNA together with the proteome associated with nascent RNA in cells that are undergoing accelerated RNA decay or not, and identifying what proteins are there by mass spectrometry and seeing, do we see a shift from sort of the normal resident nuclear set of RBPs to those that have moved in from the cytoplasm. I'll just sort of close this section by saying that we assume that this pathway that we think is related to cell stress sensed by removal of RNA from the cytoplasm is being used probably during a number of viral infections, but often pathways that are triggered by viruses we think are there for other reasons as well. And so we assumed that surely there are cases, if this is sort of a normal cellular response to removal of RNA, where non-viral stresses might also trigger its activation. And indeed, Chris Duncan-Lewis found such a pathway and saw that this is operational during the early stages of apoptosis. And so this RNA decay, was discovered by the Friedman lab, one of the former lecturers here, as a signature feature of the very early stages of apoptosis before there are gross nuclear changes. There is a nuclease that is released from the mitochondria that triggers specifically messenger RNA degradation in the cytoplasm. And what Chris found is that, like in the viral situation, this causes RNA binding protein relocalization to the nucleus in these cells and a selective repression of Pol2 activity, but not Pol3 activity, which again is something that we had seen in the viral context as well. So it's messenger RNA decay triggering messenger RNA or Pol2 transcriptional repression. Okay, so that's the first pathway, one in which RNA destruction itself is detected. But I told you that that there were two pathways involved, that there was also a control of the general protein abundance of transcriptional regulators. And this pathway we found when we went back and repeated these TMT-based mass spec experiments, but in the context of infection. So this pathway only manifested in infected cells. Expression of the viral nuclease alone was not enough to trigger it. But when we repeated these same experiments in infected cells using mouse fibroblasts infected with either wild-type MHV68 or the SOX mutant version of MHV68, we saw the same RNA binding protein shuttling phenotype that we had seen in just the MuSOX-expressing cells. But in addition to that, we saw something else. And that something else was that there was a generalized decrease in many of the subunits of RNA polymerase II itself. So Pol2 is a 12 subunit enzyme. And what we observed is that 7 of the 12 subunits of RNA polymerase II are depleted in cells infected with wild-type MHV68, but not the SOX mutant. And they're not just relocalized, they're selectively depleted in the nucleus and not from the cytoplasm. One of the interesting things about this for us was that Pol2 has 7 subunits that are specific to Pol2 and 5 subunits that are shared with the other polymerases in the cell, Pol1 and Pol3. The shared subunits, their abundance was not changing, so something about infection was specifically depleting the Pol2 specific subunits. And we thought that was really interesting. A number of viruses are known to downregulate Pol2 activity and target Pol2 itself. Generally, people are looking at the large subunit, the catalytic subunit of RNA polymerase II, RPB1. So this is really the first demonstration that viral infection could in fact lead to depletion of multiple, but not all, Pol2 subunits. This subunit depletion, which we see also in KSHV infected cells, and I'm showing you here, occurs through accelerated protein turnover. And so what we're doing here is we're taking B cells, BCBL1 cell line, which is latently infected with KSHV, and we're looking at what the half-life of these proteins are in the cells in their latent state when there's no host shutoff happening versus when we reactivate them to enter the lytic state. And so as you can see here, I'm showing you one example, which is the RPB2 subunit of Pol2. It's a Pol2 specific subunit that if we block, if we treat cells with cyclohexamide to basically block new protein synthesis and then track turnover of the existing protein, you can see that the protein in the reactivated cells is turning over more rapidly than the protein in the unreactivated cells. So there's accelerated protein turnover, and we see this for the Pol2 specific subunits, but not for the shared subunits. So we think that there's something that is activating a protein turnover of these Pol2 specific subunits in infected cells. So as I mentioned, there are 7 different Pol2 subunits. So is the virus having a strategy to somehow promote turnover of all of these 7 subunits separately? Or alternatively, is it possible that it is targeting a single subunit, and then often when you have protein complexes, their stability is dependent on the formation of that complex itself. And so if you remove a complex, a subunit from a protein complex, that can lead to destabilization of the other members of this complex. And so we had the hypothesis that perhaps it's just a single subunit that might be targeted, and then you have co-depletion of a subcomplex of proteins that are all the Pol2 specific sort of subcomplex of that polymerase. And we think this is probably the case because we could do this by selectively depleting the large subunit, RPB1, in uninfected cells, just using siRNAs. And as you can see here, that when we deplete the large subunit of Pol2, we see co-depletion of RPB2 and other shared subunits that are specific to RNA polymerase II, and we see much less co-depletion of shared subunits like RPB5. And so our hypothesis is that the virus is targeting a particular subunit or eliciting a stress that is targeting a particular subunit of Pol2 for destruction. This is leading to co-depletion of other Pol2 specific subunits, contributing to the transcriptional shutdown that we see in cells. So just to summarize what I've shown you in this first part here is that during gamma herpesvirus infection, there is a large-scale depletion of messenger RNA from the cytoplasm that is initiated by this broad-acting messenger RNA-specific nuclease called SOX. This RNA decay phenotype signals in at least 2 different ways from the cytoplasm into the nucleus to cause repression of cellular transcription by RNA polymerase II. One mechanism is the release of RNA binding proteins from these degrading RNA fragments. And the second mechanism is, we think, I didn't show you this data, but a proteasome-dependent turnover mechanism specific to subunits of Pol2 that are not shared with the other polymerases. Okay. So you might be wondering, well, these viruses are double-stranded DNA viruses. Their transcription depends on RNA polymerase II, just like the host does. So how is it that they are able to get around this phenotype? Obviously, they do, since they're very successful viruses. And so I'm going to turn my attention now to sort of the second question of viral gene regulation. So of course, while host transcription is being repressed during lytic infection, particularly later in lytic infection, viral gene expression and many other viral processes are humming along really nicely in the nucleus in what are called viral replication compartments. And so these are basically subcompartments of the nucleus that they're not membrane bound, but they concentrate replicating viral DNA, but exclude host DNA, all right? The viral DNA in these replication compartments is largely thought to be unchromatinized and accumulates to hundreds to potentially thousands of copies in here. I think of it as a really chaotic place because the DNA lacks the set of organizational principles that are used by the host DNA, and yet on that very chaotic replicating DNA sort of milieu, there are some really sophisticated gene regulatory processes and just DNA processes that are happening, including viral DNA replication. Transcription is happening off these DNA. The viral DNA is getting packaged. The localization of the viral genome within these compartments is what we think protects the viral genome from the transcriptional repression that is levied on the host genome. So this provides a protective environment where the remaining Pol2 from the cell is recruited very strongly to viral replication compartments, probably because of the open nature of the viral DNA. Non-chromatinized DNA is like a sponge for nucleic acid binding proteins, and so this helps the virus recruit factors from the rest of the nucleoplasm that it needs to coordinate its own gene expression. And what I'm going to tell you about today is how in that recruitment process, there is latent infection, very specialized viral transcriptional regulators that help coordinate the activity of Pol2 on promoters. And we found that actually connects each of these processes in some surprising ways. Right, so let me first remind you that for all DNA viruses, it doesn't matter if you're a bacteriophage or a herpes virus, you break your gene expression down into two general categories. There are the sets of viral genes that are expressed prior to the onset of viral DNA replication. These are called the early genes. Early gene promoters, by and large, look a lot like host promoters. In fact, many of these were used as model systems to understand the basics of how eukaryotic promoters act, how they assemble pre-initiation complexes to initiate transcription. So it's thought that they assemble transcription complexes, pre-initiation complexes, using the core general transcription factors and promoter elements, very much like many of our own promoters do. Late genes are the set of genes whose expression initiates only after the virus starts replicating its DNA, and this makes sense. These are things like capsid proteins, et cetera, that the virus doesn't necessarily need until there is replicated DNA to package into new capsids. These genes are the ones that are being expressed at this particularly chaotic time in these viral replication compartments where there is a lot going on in the DNA, and their expression strategies are unique compared to viral early genes and compared to host genes. First of all, they have incredibly simplistic promoters that have little more than a modified Tata box, and their transcription is coordinated by a complex of virally encoded proteins, or 6 proteins in the beta and gamma herpes viruses called the viral transcriptional activators that form a complex that directs transcription off of these late genes. We've studied this complex now for about 10 years and mapped its general organization. It's a pretty fascinating complex. That's a topic for another seminar. I'll just tell you that, for example, one of these proteins is a mimic of cellular Tata binding protein, not at the sequence level, but we think structurally and biochemically it mimics TBP, and it does something extra that TBP doesn't do, which is that it sort of acts like a bacterial sigma factor in that it can bind directly to Pol2 that is recruited into these factories and bring it to viral promoters. So TBP does not make direct contacts with Pol2 like this viral version does. All right, so we wondered though, despite the fact that we have a pretty decent idea of the organization of this complex and the promoter elements that are involved, we don't really know how it's regulated and what might participate in triggering its activity or enhancing its activity within at late times post-infection. And so this is the work of a current graduate student, Chloe McCullum, who reasoned that what she wanted to do was figure out what are the sets of things that are there, sort of at the scene of the crime. What are the cellular and viral factors that are localized to these late gene transcription complexes, to these late promoters, at the point in time where late gene expression is happening? And this sounds sort of simple, but it's actually not so simple because transcription complexes are inherently really dynamic. So you generally can't study them using sort of standard IP type assays. You're not going to be able to see these interactions. They're just happening in a snapshot in time, moving, shape-shifting a lot as they're going along. And so she reasoned instead she wanted to use a proximity labeling approach, which enables you to capture sort of transient interactions that are happening at a particular snapshot in time and place. And so for this, she used a promiscuous biotin ligase-based approach. So she's using a biotin ligase called TurboID here, which she fuses endogenously within the viral genome to one of the late gene transcriptional transactivators. And what this enables you to do is if you flow biotin into cells, this promiscuous ligase will biotinylate all of the proteins within a small nanometer radius of where this complex is. And she does this at the time where late gene transcription is happening. And then you can lyse the cells, and because biotin binds streptavidin very tightly, you can pull out all of those biotinylated proteins, and after stringently washing them, identify them by mass spectrometry. All right, so I said that like it's sort of simple, but in fact it took a ton of work to set this system up and be able to get rigorous data that we found to be believable. It was probably a year or more of trying different locations for the tag, different levels of expression, the different controls that you might layer in. So just be aware, it's This was a ton of work. Nonetheless, Khoury did this, and what she looked for, she had a number of controls built in, which I'm not going to go over, but what she looked for were proteins that consistently were found in association with this complex but not any of her negative controls, and she came up with a list of 45 proteins. These satisfyingly encompassed all of the known components of the late gene transcription factor complex and Pol2. Everything that we knew should be there was picked up by this screen. This 45 list of proteins, though, doesn't tell you whether any of these are actually involved in late gene transcriptional regulation. So she then needed to run these through a series of functional assays to test whether or not they were important for this transcription process. And for this, she needed to do some additional tool building in which she engineered on the KSHV genome in the backbone a reporter that was driven either by an early gene promoter or a late gene promoter. So if she depleted each of these, then using a CRISPR screen, she could ask whether their depletion selectively impacted late gene expression or not. Was it functionally involved? And then she took these hits from the CRISPR screen and ran them through a second round of siRNA screening, just as an orthogonal approach to make doubly sure she was getting accurate hits. All right, she did all of this, and again, picked up every single known factor that was required for late gene transcription. That was great. And exactly one new hit. You can imagine she was very nervous at this point in time. This was one of those meetings in your office where you're spending a lot of time trying to say, it's going to be okay, because really when you're doing screens, what you're hoping is that you're going to get one true hit, and you have one hit. And she's like, but what if something else tells me that hit isn't right, and I've done all of this work for years, and nothing comes out of it? It ended up being okay, because although this one hit was a protein that we did not expect at all, it turns out that this protein is quite interesting. So this is just to show you the data for this hit. The hit is a protein called ORF29. This is the late gene reporter in the virus, where you can see that depletion of ORF29 resides with all of these other viral late gene transcriptional activators, and that you have a strong reduction in late gene signal. If we do it with the early gene reporter, similar to the VTAs like ORF30, there is no repression. So this is a protein where if we deplete it, we deplete late gene expression, but not early gene expression. So it's the phenotype that we're looking for. But the protein was confusing to us because it has nothing to do with transcription as far as we can tell. It's the catalytic subunit of the viral packaging motor, the terminase, which is one of the most conserved proteins in all herpesviruses and has very clearly established roles in packaging viral DNA. No reason to think that it was participating in the transcription potency on these genomes. And so Khoury then went and triple checked herself, made an ORF29 mutant in KSHV, as expected, because if you don't have the terminase, you cannot package viral DNA in viral supernatant transfer assays, which is a measure of production of new infectious virions. You get basically the virus is completely dead. So indeed, getting rid of the terminase prevents viral production. And then she went and looked using a collection of antibodies that we have against different viral late-produced proteins or early-produced proteins, could see what was happening to these endogenous viral factors. And indeed, she could recapitulate that late proteins like K8.1 were significantly reduced in the ORF29 stop virus, whereas early viral proteins taken at the same time point were not reduced at all. All right, so what is it that getting rid of the viral terminase might do to enhance gene expression late in infection? Well, let me remind you how viral DNA packaging happens in herpesviruses. These are icosahedral viruses. You have this portal through which the viral DNA, which is replicated as at least partly in a rolling circle mechanism to form these long concatenamers that are punctuated by terminal repeats. The terminase complex binds these and in an ATP-dependent way basically pumps the DNA into these capsids. The pressure is quite impressive here. For other herpes viruses, it's been measured to be a near liquid crystalline density. This is an incredibly powerful motor in order to be able to get these in. And then it also has a nuclease activity that subsequently cleaves at a terminal repeat once you have a full genome packaged to then allow the portal to seal and the viral particle to undergo the rest of its maturation process. So one of the things that we thought was, well, perhaps if you're preventing this process, you're messing up the packaging reaction, you're going from a chaotic environment to a really chaotic environment. And this is just creating sort of, I don't know, too much something in the replication compartment. And it's not a direct effect, but it's an indirect effect of failure to package your DNA. We could test this hypothesis because a couple of years earlier, a former postdoc in the lab, Alison Didichuk, had identified another critical packaging factor for KSHV called ORF68, whose activity is essential for packaging, but is unrelated to the terminase activity. And as far as we can tell, it doesn't have any catalytic activity on its own. So we can block packaging by mutating ORF68 and see if you get the same reaction. So this is just showing you again, you get rid of ORF68, you produce no infectious virus particles because you don't package your DNA. However, if you get rid of ORF68, you express viral late proteins like K8.1 just fine. So stopping packaging does not have the byproduct of reducing gene expression at late times. There is something specific that the viral terminase is doing for this. So this is my last data slide. What Khoury wanted to determine was, is it the catalytic activity of the protein that is necessary for this function? And so what she did was she went and made point mutations in the Walker A or Walker B motifs important for ATP binding and hydrolysis for the packaging reaction, and in the nuclease domain that is important for clipping that DNA to finish the packaging reaction. And then asked, can any of these mutants complement, when she transduces these into cells, a virus that lacks ORF29? So she compared the mutants to wild type. And I will just summarize the data for you here because basically the mutants all look the same, which is that if you mutate any aspect of the catalytic activity of the protein, unlike the wild type protein when transduced in, which complements fine, you are unable to complement this defect in expression at late times. And so this says that the catalytic activity of the protein is indeed critical for potentiating gene expression late in infection. All right, so let me bring this together for you and say why we think that this is interesting and important. First of all, I mean, for a long time, it has been known for all DNA viruses that this act of DNA replication is required in some way to license transcription of genes late in infection. But what we're showing now is that indeed it is not just this critical activity, but then downstream of that, the packaging reaction also somehow feeds back to potentiate gene expression as well. And how might this be happening? We have a couple of ideas. Because the ATPase activity is required, one possibility is that the terminase is functioning sort of in, we don't think it's just a scaffolding factor, but maybe it's sort of scanning and clearing the DNA to make it more easier to transcribe. Alternatively, we think that one possibility is that the packaging reaction itself is you're twisting the DNA into the capsid, you're creating torsional strain on DNA. And torsional strain is the same thing that happens when our own DNA is being replicated. You need things like topoisomerases to relieve that torsional strain, otherwise it will cause transcriptional inhibition. So we favor the idea at present that perhaps the terminase, through its packaging and nuclease activity, is relieving the torsional strain that is building up during the packaging reaction, which is necessary to allow transcription to continue to happen on those newly replicated genomes. This also is interesting, I think, because it suggests that if inhibitors can be developed against the packaging complex, so something like what has been done for CMV, that those inhibitors might be potent not just because they block packaging, but because they also poison late gene transcription, which is critical for completion of the viral life cycle. Okay, so I'll end there and mention that the first part of my talk was carried out largely by these individuals boxed here. These are former Ph.D. students who had started the work. The Pol-2 protein subunit turnover work is largely being carried out by a current Ph.D. student, Leah Goulias. And the late gene work, the story was largely carried out by Chloe McCollum with a lot of help from former postdoc Alison Dudachuk, who is now at Yale and helped us build a number of the tools and get the system set up. And of course, always grateful for our funding, and I'd be more than happy to take questions. I'll just leave up our model there to help you think of any if there's time. Thanks.
We do have a question online. I'll take that first before we go. So our friend Howard Young up at NCI Frederick asks, do interferons play any role in your data?
Do interferons play any role in my data? I guess I wonder which, I'm guessing he's talking about the first part.
Yeah, this question came in pretty early, yes.
Yeah. So an idea is that one of the roles of host shutoff is to restrict production of interferon-stimulated genes. And that's been pretty well established for a number of host shutoff factors in these and other viruses. And so counteracting the interferon response is an important role of host shutoff. Although I wouldn't say that we have any evidence that interferons themselves are preferentially targeted, that it's just interferon-related genes are captured in the set of genes that are targeted for destruction.
Okay, go ahead.
That was great science and a great presentation as well. I have a question about, on your model slide there, of the poly-A binding proteins being imported into the nucleus. Could that potentially inhibit translation of viral genes, or could that benefit translation of viral genes? Because I'm not remembering my translation, whether or not the poly-A binding proteins bind to the poly-A tails in the nucleus or the cytoplasm of new transcripts.
Yeah, you're not remembering that because it's not been well established.
Okay, okay.
But the thinking is that it probably binds them in the, so it's normally a shuttling protein. It probably binds them in the nucleus and then is escorted into the cytoplasm and then cycles back into the nucleus. Yes, I would think that PAB is important for viral translation, and we've looked at where it is in the nucleus, and in fact, you can see it in viral replication compartments as well. So we originally thought, well, maybe it's excluded, and this is one of the ways in which viruses escape, but it's possible that it's actually used by the virus in the replication compartments in processing those RNAs so that they can be exported efficiently and translated efficiently as well. So the virus needs it too.
Great.
Yeah, that was lovely. Thank you. I could ask whether phage do the same thing, but I don't know whether anybody's
and they
looked.
probably do. There's remarkable similarities between phages and herpesviruses.
So in the last bit, the torsional release, do you think you need the package at the end to, is that what it's fighting against, or if you didn't have something going on there?
If there was no packaging, would you still need? That is an excellent question, and the way that we could address that would be to make the double mutant of an ORF68 to block packaging and the ORF29 terminase and say, in the background of no packaging, do you still need to relieve the torsional strain? I hadn't thought of that experiment. I'm going to totally bring that up to my student. Thank you. She's probably already thought of it, but.
There was a protein, PAT-L1.
Yeah.
In your third land in your
Yeah.
mass spec, what is that, and what do you think is going on with that protein?
Yeah, so it turns out that there's a number of factors involved in RNA decay, and that's one of them that tend to shuttle into the nucleus, in particular things involved in decapping, and we delved into that a little bit when we were first working on these data. And I'll say I don't have anything satisfying to tell you as to what the outcome of that is, except for we noticed that there are a number of things that are involved in messenger RNA decapping and some other regulators of RNA decay that happen to shift localization in the face of RNA decay. So, yeah.
You have a question up here?
I'm asking, is there any modification of the proteins which occurs after this infection? Why I'm asking the question is following.
Can you get a little closer to the microphone?
Okay, yes. Back in 1967 or 68, it was found that, at least I was a participant, that the T4 phage, when it attaches the E. coli cell, the RNA polymerase immediately gets inactivated for
Yeah. Yeah.
That is because ADP ribosylation of the sigma factor takes
Yep.
place. And then that modified one only transcribes the T4 DNA. So I think this must be very important that some modification takes place that then they are active with respect to the alternative.
Yeah,
no?
yeah. So
That
you're
has-
asking whether or not there's any modification of the Pol2 subunits that,
Yes.
that undergo decay.
Decays.
Yeah, that's a great question, and the simple answer is I don't know, but we suspect that there's probably ubiquitination. We think that it's a ubiquitin-linked turnover mechanism.
Maybe.
We don't know what is the ligase or ligases that might participate in that, and the hypothesis is that, well, there's two possibilities. One is that there's a viral protein in particular that is targeting these for turnover, and the other hypothesis is that it is linked to a cell stress. There are other cell stressors, DNA damage, collisions that happen on DNA, things that will also trigger removal of polymerases through proteasome-mediated turnover. So is the virus activating a stress that is promoting that? But presumably it's through a modification that
Yes,
triggers the turnover.
modification, and you have the conformational changes or whatever, so the interactions are not possible and so on and so forth. Okay.
Yeah.
Okay, maybe we can squeeze in two more before we go.
Hey, Britt.
hello.
Wonderful talk. How are you? I have a question following the PABC1 translocation and nucleus. So you show data in mouse herpesvirus 68 about KSHV. We know that KSHV encode PAN,
Yep.
which is
Yep.
polymerase nucleus, the RNA, highly accommodated nucleus. Can it kind of sponge the incoming PBC1?
Yeah, which is something that also Joan Stites has talked about, this idea of, okay, so you have this hyperabundant polyadenylated nuclear RNA in KSHV infected cells. It is binding the PAB-C that is coming in from the cytoplasm. And so the question is that in the context of KSHV, that's probably one of the things that maybe is helping retain PAB in the nucleus. Certainly it's influencing, presumably, PAN's stability there. We don't think that that's what's bringing PAB into the nucleus. We think that it's the RNA decay that is helping move it into the nucleus. PAN is probably serving as a sponge for it in the nucleus. And whether that means that it's free to do different things in an MHV68 infected cell that lacks PAN compared with a KSHV infected cell, I think is an interesting possibility that I don't know the answer to. Yeah.
Thank you.
Beautiful talk. So, you spoke to the chaos of replication, and I think packaging's been kind of more of a black box, and so you have replication, late gene expression, and packaging, which seem to now be linked.
Yeah.
One question I had, maybe a simple hypothesis for the terminase defect is if you're not packaging the genomes, do you just have more genomes overall, and you have a kind of a distribution of POL2 in these late complexes, and that's why you're getting late gene expression?
Yeah, I think that was a possibility that we considered, but that I think is disproven by the fact that we can block packaging with the ORF68 mutant and not impair expression of late genes, which suggests that just having more DNA around, it is not that you've got a limiting amount of Pol2, which is probably there sampling the open DNA all over the place together with other nucleic acid binding proteins. Yeah.
Excellent. Eric, any closing thoughts?
Thank you very much for the great
All right,
seminar.
thanks for the great questions.